Thanks, Barbara, and thanks for inviting me today to talk about the LungMap, or the Lung Master Protocol, which, as you see, is a phase 2-3 study in the second-line setting for squamous cell carcinoma of the lung. But it's really much more than that. As you'll see, we're hoping that this will be a new paradigm for how to develop these drugs and get them approved even faster. So we call it LungMap, and the first thing I should say is I'm here representing a large collaborative effort. It really is a major enterprise of the SWOG and really all the cooperative groups and the NCTN working together. We have support from the FNIH, very closely aligned with the National Cancer Institute, as you'll see. Many of the ideas for this actually came from the FDA, and I'll talk about that. And then, of course, Friends of Cancer Research and many others, many individuals. So I'm really honored to be here to discuss it. Well, this all comes out of everything we heard about this morning, very nicely discussed by Bruce and others, Mia, that we really have taken lung cancer and made it a disease where we're focusing really on smaller and smaller pieces of the pie. And we have 3 or 4 driver mutations you heard of. Most of those tend to be, by the way, in non-squamous carcinoma, in people who have smoked very little or not at all. And most of those patients will become resistant. So what about the other 80%? What about the squamous patients, many of whom have smoked? Well, we need to continue to find new and better therapies. Well, the science is there. Certainly the TCGA project has given us some hope. We're sequencing large numbers of squamous tumors, for example, have produced data such as these from Dr. Hammerman and colleagues in Nature in 2012. And I'm not sure how I point. Maybe this is a pointer. But what you can see is if you look here, there are specific mutations. Are they drivers? We don't know for sure. But there are specific mutations. And one that I would point out is the 4th down PIK3CA, PI3 kinase mutations. They're there. They're about 15%. So if you wanted to do a trial and test whether that is a driver and test a drug or a series of drugs against that, you'd have to screen several hundred patients to have enough for a clinical trial. I can tell you that's impossible to do at any one center. It's impossible to even do at 10 centers. It's very frustrating for us, the caregivers, but it's more frustrating for the patients. Because the patients, we heard how valuable the tissue is. They get their tissue tested for one trial. They're not eligible. 2 weeks go by in a disease where the median survival is just a matter of months. So there needs to be a better way to find the right drug for the right patient at the right time. And a lot of this, of course, came out of this committee, the Policy Forum. I still remember, I guess it's more than 5 or 6 years ago now, at one of the meetings when John Niederhuber came, and we talked about how could we reform the clinical trial system. And many in this room are there. And of course, John Mendelsohn chaired this committee, and it's a seminal report basically looking at the clinical trial system. How could we make clinical trials more relevant? How can we make it so that there's more science in the trials, that drugs could get approved faster? So that was the challenge of the IOM report. So the whole master protocol sort of came out of the desire to do something, to take the recommendations from this report that came out of this group to see if we could do better for our patients. Well, lung cancer was a perfect place to start. We because of the EGFR mutations now, 10 years since the discovery, and even a little bit before that, lung cancer became a disease where tissue was being obtained. That wasn't the case, by the way, 15, 20 years ago. You'd be hard-pressed to find even fine needle aspiration to study a tissue in lung cancer. But because of work like what Bruce described and others, more and more people were obtaining tissue, not the resected specimens, which are all well and good, but the specimens from the refractory patient, the patients who's had chemotherapy and radiation therapy, because after all, what's left and growing, these are the cells that we want to kill. So rebiopsies and biopsies in lung cancer became much more common, and that's important as we talk about the master protocol. And then, of course, there were, as you heard about, there were new clinical trial ideas, and we call them the umbrella or the basket trial, and both very important. Of course, the basket trial was critical for developing imatinib in so many different tumor types. The idea being that you take a molecular abnormality, no matter what the tumor type, and you test the drug against all of those. And of course, a BRAF study is another example, the NCI MAX that we already heard about. But what we're focusing on now is an umbrella, whereas an umbrella is taking one specific tumor type, or in this case, refractory lung cancer, and treating patients with different options. And I'll show you that. And previous examples of this that have been discussed before at the forum, the BATTLE trial, which we were very involved with, the iSpy trial, and of course our current iteration. Just for those who want a little review. We actually presented this at one of these forums several years back. The battle trial was won early on in the process. It was an early umbrella. It was a phase 2 umbrella where we did core biopsies on patients with lung cancer. Here's a CT-guided core. We tested for a panel of 14 different markers across 4 different disease groups. And then we used a Bayesian randomization to place patients on 4 different drugs. And this was an early iteration. A lot of the feasibility and a lot of the fundamentals of how to do this were worked out in this study. But then we wanted to do even more. We wanted to perhaps go all the way to phase 3. That was only a phase 2 marker discovery program. So how did we do this? We want to address the issues of modernizing the process, as I mentioned. We wanted to work in a collaborative way with all of the different stakeholders. So this all really began around 2011 when I got a call from Ellen Siegel, and she asked me to chair a group for her Friends Brookings meeting that year to look at this issue. And we published a white paper, and this white paper really was the basis for how to develop a master protocol, and we decided to focus on squamous cell lung, and you can see the committee members shown here. Well, of course, we weren't working in a vacuum. At the same time, the NCI task force, the lung steering committee chaired by Fred Hirsch, was also having meetings to discuss the same thing. I happened to be on both groups. When I realized that there were 2 parallel efforts going on, I said, why don't we all work together and try to do this trial through the NCTN, through the new national system. What a great demonstration case for how the new network's going to work. Of course, the NCI was talking about earlier stage trials, the Alchemist, which was mentioned already earlier, and then advanced disease. So we decided we'd pool our forces on an advanced disease master protocol. And just to show you, these are some of the other NCI iterations. I borrowed this. I was at the NCI last week and working with Barb and Jeff Abrams and others. You can see the NCI has many different trials using this sort of partnership, public-private partnership in many cases, to run their studies. So again, this is just, I can't emphasize enough that this really is, many working together. Janet Woodcock and Rick Pazder have been very involved, and they came to us and challenged us to do this and have been giving us advice along the way as much as they can in a protocol that hopefully will be used for phase 3 approvals. A lot of pre-competitive work between companies, as you'll see in a moment. But you can basically see that we have the Friends, the FNIH, which is where a lot of the private money is put through as it gets back into the cooperative group structure, the NCI, and of course the FDA. So here are the main hypotheses of this study. As I mentioned, we wanted to use the National Clinical Trials Network, newly minted. I guess it's only been in effect since March or so of this year, to provide a mechanism for genomic screening of large homogeneous cancer populations, to find those 5%, to find those 10%, to do it in a one-stop platform, excellent for patients, good for the development of drugs. We want to assign and accrue simultaneously to a multi-sub-study master protocol comparing new targeted therapies to standard of care. No placebos here, standard of care. It's the docetaxel that Adrian showed us at the beginning, so we need to do better based on designated therapeutic biomarker drug combinations and improved genomic screening for clinical trial entry and improved timelines for drug biomarker testing, allowing for inclusion of the maximum number of patients to allow for drugs to hopefully be approved more quickly. And that's the goal, to identify and quickly lead to approval, safe and effective regimens for patients with this disease. So here's how it works in a nutshell. Pretty simple. Each of the arms is modular. Each arm, patients are consented into the umbrella. The tumor is collected. There's a great deal of private money in this. It's a $150 million trial. 3/4 of that money is coming from the companies that are providing the drugs, and we can talk about how we selected those. So that money is not only providing the sequencing so that it can go off to Foundation Medicine in this case, but also there are resources for new biopsies. So if we need a new biopsy, there isn't enough, that's paid for. And that's huge because we're getting this trial throughout the United States, through Canada, to community sites that might not have been doing this in the past, perhaps because of resource constraints. We then have the genomic screening, which has to be done within 2 weeks. Foundation Medicine was chosen through an RFA, and then based on the results, we assign the patients to one of 5 arms. On each of those arms, the patient's either receiving standard of care or an investigational targeted therapy, simply like that. So here's the design. The umbrella is shown up here. Here's our umbrella. There are 4 arms based on the biomarkers. We used the science, the best science that was available. And then we realized we'd only be able to assign about 50% to a specific biomarker group. And then we have a 5th group, which we call the non-matched arm. for which there isn't a driver or a hypothesized driver mutation. And for that, actually, you'll see we're using immunotherapy. In each arm, the patient's getting chemotherapy, in most cases docetaxel, versus the drug that's being tested. In one arm, we're actually using erlotinib as our control, also approved in second-line squamous lung cancer, versus erlotinib plus, in this case, an HGF inhibitor. Here's our leadership team shown here, working very closely with Vali Papadimitriopoulos at MD Anderson, David Gandera, who chairs the SWOG Lung Committee, Fred Hirsch, who I mentioned had come through and is with the NCI committee and the translational chair, and Mary Redmond, who's done a wonderful job with our statistical design. We have a drug selection committee. We try to be transparent. We try to include members from all aspects of the stakeholders except for people currently with industry, because we didn't want to have them biased, and also we were looking at confidential information on many drugs. That was quite hard, but we found people, I don't see it, like Gwen Fife and others who had worked with many companies, but were now consultants. And so we had some expertise there. And of course, as a non-voting members, and to keep us honest, we had members of the different, the FDA, actually even Janet Woodcock was on the committee. We discussed this with more than 20 companies, multiple agents from each company, and we picked only 5. These are the 5 that we went with. It was very difficult. We based it on phase 1/2 data. There had to be some evidence that these drugs were active in this setting and safe. You know, the best drug in class might not have been the best drug for the trial because there were no data yet on it through phase 1. There also had to be a biomarker. I'll tell you, the most frustrating thing for me is the biomarkers still are not that well developed. We probably need more battle-type trials. We need more of a farm team to develop the biomarkers in the drug. But we found 5 that we felt were very reasonable to go forward with. An FGF receptor inhibitor from AstraZeneca. This is a small molecule. They've actually tried to do trials, the company on their own, and had a great deal of trouble finding patients. I can tell you by the time you find patients for this, this is probably 5 to 10% of the population. Those patients are probably too sick to go on the trial if you do it through the normal mechanism, because it can take so long to find the patients and to identify them. A PI3 kinase, beta-sparing PI3 kinase inhibitor from Genentech. This is a PD-L1 antibody from MedImmune. This is the non-matched arm. Palbociclib, which is, of course, a quite exciting CD4/6 inhibitor. Some very good results now in breast cancer. There have been some responses in lung cancer. And rilotuzumab, which targets the whole c-Met pathway. Bruce had mentioned that there was some evidence that that was important in lung cancer. I do have a slide on each of these. I don't think it's that important to go through, but I'll just say that, you know, the immune checkpoints, phenomenal results in lung cancer, still in only 20% of the patients. I've been criticized, why is this the non-matched arm? Because we don't have the right marker for it yet. But I'll tell you, we're going to learn markers from this because we're going to have 1,000 patients all sequenced, and perhaps we'll be able to figure out what the marker is. We'll give Neil his sequencing marker. And then sub-study B, this is the beta-sparing PI3 kinase inhibitor from Genentech. I think it's fair to say these agents are quite exciting preclinically. Clinically, I think we've been a bit underwhelmed in the lung cancer arena so far as to their activity. But we're going to ask the question now with a specific group of patients, biomarker profile, do they respond? Sub-study C, this is the CDK4/6 agent from Pfizer. Again, still very early, but I'll show you there's a phase 2/3 design so that if it doesn't work in phase 2, we pull it out and we go to the next drug. This is the FGF receptor inhibitor. And then finally, this is the c-Met inhibitor. So 5 drugs. Now, we needed a platform. And when we were designing this, we were using NCI funds. We felt it was very important we do a transparent RFA process. So working with experts, Mickey and others who are here, we actually had an RFA, and 3 or 4 companies came to us and saying, we want to bid to do your profiling. We ended up choosing Foundation Medicine. We have a competitive price with them, and they are so far delivering quite nicely, where they are doing their same platform that they do commercially, except they do it they don't prepare the full report. They just provide us the marker information that we need for the 5 arms, and within 10, 15 days, 10, 12 days, they give us these results. Now, as we heard this morning, not everything's going to be a gene. There is protein assays that need to be done. And we have an IHC, immunochemistry for MET, and that's being done by Clarient in California. So there's a coordination between the 2 groups with the same tissue. In fact, here's how it works. The tissue's obtained at the site, either using an archival biopsy or a new biopsy if felt necessary. A pathologist has to look at it at the site. By the way, we can't take the pathologist out of this whole process. There's a push at some centers, just send it to the sequencing center. You can't do that. Someone's got to look at the tissue and tell you how much tumor is there and how much other cells. Then it goes to Foundation Medicine, where they then divide it up, and some goes to Clarient, some goes to Foundation. Clarient sends the results to Foundation, so the SWOG Statistical Center just gets one report, and then the sites are notified of their sub-study. There are some issues. I just bring this up because we'll discuss this more, I guess, later in the meeting. We've had other meetings here at the IOM about this, but there's some big ethical issues as well here. We're doing squamous lung cancer for which there is no standard of care that one should test and do sequencing. However, our initial thought was we'd have a pre-screening component. We'd run this test before the patient even failed their frontline therapy to help accrual to the trial, and then we'd return results to the patient after progression on the study. However, the IRB, and I'm learning that every IRB can view these issues differently, and I think it's going to be important for groups like ours here to try to set some standards regarding this. The central IRB said the full results of the molecular analysis should be presented to the patient prior to enrollment. So they wanted us to give the patient the full report even before we put them into one of the 5 sub-studies. And we talked about it. We couldn't fight that. We don't even know what to do with the data. I'm not sure how you would explain that to the patient. But I just present this because these are issues that are coming up. The public is being increasingly aware of the availability of the testing and the implications on family histories. Even if you do a Foundation Medicine test where you don't have normal sequencing, you can still figure out, for example, BRCA1, BRCA2. There are some implications there. And the patient may have privacy confidentiality concerns. So actually, what we do now is we don't pre-screen right now. Patients get all the information prior to enrollment, and we've created educational materials to at least instruct the physician how to do that with a patient. Just an aside. So just as I finish up, here's how it works. I know some of you might want to see the statistical plan. So there's a phase 2-3. Many of these drugs, and I'm sure you'll ask me on the panel, some of them are long shots. They're the best we could find, but they might not all make it. But we only need 55 progression-free events in each sub-study before we take a look and establish futility. So, and then if that looks good, if it passes the futility boundary, then it can go on, and these patients all count in the final analysis, which occurs over here. The primary endpoint of this trial is a co-PFS overall survival endpoint. We had serious negotiations with the NCI in this one. We wanted to do PFS because this is only second-line squamous lung cancer, and we know patients will go on to get third, fourth, and fifth line, which could skew an overall survival result, so we agreed to do both. So just some of you might want to see, what is the stringency? ASCO had the meaningful outcomes paper recently. I was actually part of that myself. So these are going to be meaningful outcomes if this trial hits, and I'll show you why. In Plan A, which was the plan that we proposed, the hazard ratio for the phase 2 is 0.5. That means a 41% increase in order to move on to the phase 3. That's a pretty high bar. But you know what? 2 of the companies came to us and said, that's not even high enough. And the reason it wasn't high enough for them is when a company puts their drug in the master protocol, they say goodbye to all of their control over it from then on. It's all being done by the cooperative group, DSMB. They don't have a chance between phase 2 to 3 to make decisions. So, of course, there's a great financial cost in moving from phase 2 to 3. So 2 of the studies said we want to actually have the hazard ratio 0.4 or see, we want to have a hazard ratio of 0.61 or a 63% increase. That was actually done by 2 of the groups. As far as the overall survival, we only have one plan for the final PFS and overall survival, and that's a hazard ratio for PFS of 0.5 and overall survival of 0.67, as you can see here, as shown. So a 2-fold increase or a 50% improvement. You can see the sample sizes. I just wanted to show you that based on the prevalence of the marker, the sizes are different. But the phase 2s are between 100 and 200 patients, and if it goes to phase 3, including the phase 2 patients, somewhere on the order of 300 to 400 patients. So this would be the numbers of patients if it went all the way. Some of the markers will overlap. Always a problem when you do this type of work. Not sure what to do about that. We're aware of it based on historical data. Who knows what it will be in real time. The way we've decided to deal with this is we randomize whenever there's multiple markers present in the specimen with a skew towards the lower prevalence marker. Otherwise, we'd never fill those arms. So just as I conclude, so here's the final trial. Again, fresh biopsies if needed. Here's the Executive Operations Committee. As you can imagine, the infrastructure Keeping the firewall between companies, bringing the drugs in, keeping it moving is quite massive. Amazing support from David Woolley and the FNIH. As I mentioned, Ellen Siegel and her group are just phenomenal. The NCI, the cooperative groups, all of them, Chuck Blanke leading the SWOG, who we're working with all the groups, and Valley and I share the operations group, and we meet on a weekly basis. We're meeting our timelines. We started the trial in June of this year. It's now open in over almost 300 sites around the country. The thing that amazed me is some of the private groups came to us and asked if they could join, groups that don't normally participate in cooperative group studies. I won't mention who they are right now, but it just was amazing that they called us up. And also, we're working very hard to get Canada going, and that should happen by the end of the year. So basically, right now, we have IRB approvals at 353 sites. We're using the central IRB. That's good. That's something we've talked about at this forum many, many times. The one thing that, even when the central IRB is moving, there's still some steps after that to get going. But still, we have 35 sites now with at least one patient accrued, and we have nearly 100 patients enrolled in the first 3 months, and it's going forward, and we're continuing to evaluate new drugs. So finally, we were asked to present as many were at the 21st Century Cures Committee, Upton Committee, and so we presented the master protocol, and with the help from many here, I used this as a chance to really focus on the need for more biomarker development, more resources for diagnostic development, the whole public-private partnership. I cannot underestimate, I don't want to overestimate, how important that is, and important it is to work together and to do this in a pre-competitive way. And then, of course, we could still use a few more resources from the NCI. So last slide, really, we have patient materials on the web. We've used some of the extra money to try to get the word out. We're letting people know that they should test and sequence. And we actually have a website here if anyone wants to look at it. And this is one of the websites, really showing that, it's meant for patients and physicians alike. And really my final slide, that we hope that we have now developed a program where we have improved enrollment efficiency, improved operational efficiency, consistency as to how drugs are tested, each drug in an identical way, that we have a predictability of how drugs might benefit patients. And of course, the ones that benefit the most from this are the patients. And I know there's some advocacy groups here. We're working very closely with them to try to bring this to fruition. And then just my last slides, as I said, I'm one of many. It really is just a wonderful collaboration of investigators. Thank you very much.
Okay, thank you, Roy. We're going to move on to Lillian Hsu, who is going to tell us how the Ontario Cancer Institute and Princess Margaret is approaching the problem of finding the variants and finding the treatments.